Sarcopenia is commonly associated with aging. However, growing evidence suggests that diabetes and obesity can accelerate the loss of muscle mass and function, leading to what is known as secondary sarcopenia. SDT fatty rats spontaneously develop obesity and type 2 diabetes. In this study, skeletal muscle changes were examined longitudinally to better understand how metabolic disease contributes to muscle dysfunction. Muscle weakness and reduced muscle mass are frequently observed in patients with diabetes. Yet the sequence of pathological events leading to muscle loss remains incompletely understood. To address this question, skeletal muscle pathology was investigated throughout the progression of diabetes in SDT fatty rats. Different muscle fiber types can respond differently to metabolic stress. In this study, the soleus muscle, which is rich in slow-twitch fibers, and the extensor digitorum longus, or EDL muscle, which is dominated by fast-twitch fibers, were evaluated and compared. SDT fatty rats and age-matched Sprague-Dudley rats were examined from 8 to 40 weeks of age. Multiple parameters were evaluated, including muscle weight, histopathology, lipid accumulation, gene expression, circulating biomarkers, and muscle strength. Let's first examine how skeletal muscle mass changes over time. In normal SD rats, skeletal muscle mass increased progressively with age. In contrast, muscle growth was impaired in SDT fatty rats, with particularly pronounced reductions observed in the EDL muscle. But was this simply a delay in growth, or was true muscle atrophy occurring? To answer this question, muscle fiber morphology was examined in greater detail. Analysis of muscle fiber cross-sectional area revealed more pronounced atrophy in fast-twitch type 2B fibers. These findings indicate that the reduction in muscle mass reflects genuine structural changes within skeletal muscle. Interestingly, evidence suggests that pathological alterations begin even before overt muscle atrophy becomes apparent. One of the most notable findings was the early accumulation of lipid droplets within skeletal muscle cells. Adipofyllin staining demonstrated increased intramyocellular lipid deposition, while electron microscopy revealed lipid droplets accompanied by mitochondrial alterations. These changes were observed before clear muscle fiber atrophy became evident. The next question is whether other physiological changes occur alongside these structural alterations. Plasma IGF-1 levels were measured throughout the study period. As diabetes progressed, plasma IGF-1 concentrations decreased in SDT fatty rats. This reduction became more apparent during the later stages of disease progression. To further characterize skeletal muscle pathology, muscle atrophy-related genes were also evaluated. Expression levels of atrogen-1 and MUF1, two genes commonly associated with skeletal muscle atrophy, were examined. In SDT fatty rats, no marked increase in either gene was observed during the study period. These results indicate that muscle atrophy occurred without substantial elevation of these markers under the conditions evaluated in this study. The remaining question is whether these pathological changes are reflected in muscle function. As muscle mass decreased and muscle fibers became smaller, grip strength progressively declined. These results demonstrate that SDT fatty rats reproduce not only structural abnormalities, but also functional impairment of skeletal muscle. Taken together, the model exhibits key features of diabetes-related sarcopenia. Let us summarize the overall pathological progression observed in this study. During disease progression, SDT fatty rats exhibited hyperglycemia and dyslipidemia from an early stage. Intramyocellular lipid accumulation and mitochondrial alterations were observed in skeletal muscle, together with reduced plasma IGF-1 levels. In addition, selective atrophy of fast-twitch muscle fibers and progressive loss of grip strength were identified. Taken together, These findings demonstrate that SDT fatty rats reproduce multiple pathological features associated with diabetes-related sarcopenia.